Hi, today we'll talk about transcription of mitochondrial DNA and we'll look at the differences between mitochondrial and DNA transcription. Mitochondrial DNA is a very small circular DNA molecule that resembles a bacterial chromosomes. It is not present inside the nucleus, it is outside the cellular nucleus, and it has its own DNA, which is circular and does not utilize histones as compared to DNA, cellular DNA that has histones. Transcription in the mitochondria takes place within the mitochondrial matrix. This is the innermost compartment of the mitochondria, surrounded by the inner mitochondrial membrane. The matrix contains the mitochondrial DNA. It also contains RNA polymerase, which is slightly different from DNA polymerase 2. This one is it's called Pol RMT and other necessary transcription factors like TFP2M and TFAM. The matrix environment is where the transcription machinery assembles and synthesizes RNA from the mitochondrial DNA templates. Here on the left, you can see the structure of a mitochondria. What is worth noting is the mitochondrial DNA, the ribosomes inside the matrix. Both the mitochondrial DNA and the ribosomes are inside the mitochondrial matrix. Another important or noteworthy thing to remember is that mitochondrial translation takes place in the mitochondrial matrix as well. So, not only transcription, but both transcription and translation takes place in the mitochondrial matrix. This is where the mitochondrial ribosomes, which is very distinct from cytoplasmic ribosomes, are located. These ribosomes translate the mitochondrial messenger RNA synthesized in the matrix into proteins. These proteins are primarily components of the mitochondrial respiratory chain complexes and other essential mitochondrial functions. The mitochondrial matrix thus serves as a site for both transcription and translation within the mitochondria. The key steps in mitochondrial transcription. So it starts by initiation. Mitochondrial transcription is initiated at specific promoter regions on the mitochondrial DNA. These promoters are often denoted as heavy strand promoters and light strand promoters, reflecting the heavy and light strands of the mitochondrial DNA. The mitochondrial RNA polymerase, Pol RMT, recognizes and binds to these promoter regions. Pol RMT is assisted by transcription factors such as TFP2M, which simply is transcription factor B2 mitochondrial, and TFAM, transcription factor A, which is also a mitochondrial transcription factor. After initiation, we have elongation. Here, the RNA polymerase begins synthesizing RNA from the DNA template. This process is similar to nuclear transcription, but it uses Pol RMT instead of nuclear RNA polymerase 2. The elongation phase involves the addition of ribonucleotides complementary to the DNA template strand, forming a nascent RNA molecule. Termination. Transcription will terminate at specific termination sequences in the mitochondrial DNA. The termination of transcription is regulated by mitochondrial termination factors like mTORF1, which is basically mitochondrial transcription termination factor 1. So we'll just look at the key components of the mitochondrial transcription. Mitochondrial DNA, circular DNA, which is located in the mitochondria, encodes essential components for the mitochondrial function. So the mitochondrial DNA will code for Pol RMT, the mitochondrial RNA polymerase responsible for synthesizing RNA, transcription factors like TFP2M, a transcription factor that assists Pol RMT in promoter recognition and transcription and initiation, and then TFAM, which is another transcription factor that helps the compaction of mitochondrial DNA and transcription initiation. We have mTORF1, which is just a termination factor that helps in properly ending the transcription process. So that molecule is called mTORF1. Importance of mitochondrial transcription. Mitochondrial transcription is crucial for producing the RNA necessary for synthesis of proteins required for the mitochondrial electron transport chain. These proteins are essential for ADP production through oxidative phosphorylation a critical energy producing process in cells. So, understanding mitochondrial transcription is vital for insights into various mitochondrial diseases and conditions where energy production is impaired. Mitochondrial messenger RNA. So, the origin is mitochondrial messenger RNA is transcribed from mitochondrial DNA. Mitochondrial DNA is circular and much smaller than nuclear DNA. We already went through that. Transcription machinery. Transcription in mitochondria is performed by Paul RMT, which is different from RNA polymerase II used in the nucleus. Specific mitochondrial transcription factors like TFP2M and TFAM are involved in the transcription process. RNA processing. Mitochondrial messenger RNAs generally undergo less extensive processing compared to nuclear messenger RNAs. They typically lack extensive 5' capping and polyadenylation seen in nuclear mRNAs, although some level of polyadenylation occurs to stabilize mitochondrial messenger RNAs. In the structure, mitochondrial mRNAs often have simpler 5' 3' untranslated regions. They may contain untranslated regions that are used in a polycistronic manner, meaning several genes are transcribed together in a single mRNA molecule and thus processed into individual mRNAs. Translation. Mitochondrial messenger RNAs are translated by mitochondrial ribosomes, which differ structurally and functionally from cytoplasmic ribosomes. Now, nuclear DNA, the origin of nuclear DNA, mRNA, is transcribed from nuclear DNA, which is linear and organized into chromosomes. Nuclear DNA contains a much larger number of genes compared to mitochondrial DNA. Transcription machinery. Transcription in the nucleus is carried out by RNA polymerase II, along with a plethora of general transcription factors and regularity elements. RNA processing. Nuclear mRNAs undergo extensive processing, including 5' capping, splicing to remove introns, and 3' polyadenylation. Alternative splicing allows for the generation of multiple messenger RNA variants from a single gene. What's key is that in nuclear mRNA, there's alternative splicing, which allows for the generation of multiple mRNA variants from a single gene. In the structure, nuclear mRNAs generally have complex and longer 5' 3' untranslated regions and play roles in the regulation of translation and mRNA stability. Nuclear mRNAs are translated in the cytoplasm by cytoplasmic ribosomes, which are distinct from mitochondrial ribosomes. Looking at the yellow box, while both mitochondrial and nuclear mRNAs encode proteins essential for cellular function, they differ significantly in their origins, in their processing and translation machinery due to the distinct environments of the mitochondria and the nucleus. Now let's look at this question. Now that we know the differences between mitochondrial and nuclear transcription methods, let's look at this question and see if we're able to answer it. So, while analyzing the eukaryotic genome, small circular DNA molecules that resemble a bacterial chromosome is isolated. These molecules code for proteins, transfer RNAs, and ribosomal RNAs. From which of the following cellular structures did these DNA molecules most likely originate? So, the options are nucleus, mitochondria, bacterial ribosomes. I think by now everyone should know that the answer is most probably and most likely mitochondria, because mitochondrial DNA is a small circular DNA molecule that resembles a bacterial chromosome.